Hi everybody, hope you're fine. Welcome to the Brain Book channel. I am Amir, and in this video I want to talk about neuronal lineage including glia and neurons. We will talk about neurons and neuroglia comprehensively. The cells in our nervous system divided into different groups. One is neurons and another one is glia. The neurons are the cells that transmit nerve impulses along the nerve fibers to another neurons, and they are made up of bundles of nerve fibers. And glia are the cells that have a variety of functions to support, aid, and protect the component of nervous system. Exclusively, neurons are work as a cells that transmit the nerve impulses, and the glia play as a supportive cells. And as you can see here in the diagram, the neural stem cells will be divided in the two main group of cells, including neuronal progenitor cells and glia progenitor cells. And neural progenitor cells will form all kinds of neurons for impulse transmissions, and glia progenitors will form the different neurons that have a protective and also supportive effect in the nervous system. The glia are cells that are numerous as their number equals to the neuron, and they are so essential for normal development and function of nervous system. And they are divided into two main groups. The first one is microglia, and another one is macroglia cells. The microglia are totally 5% of glia population in our nervous system. And we should notice that the microglia have a hematopoietic lineage, and they are not formed from, for example, neuroectoderm, and they are not the lineage of neural stem cells. And their role is scavenging and degrading the dead cells and protect the brain from invading microorganisms. Totally, the microglia, when they want to act or may have a function, they enter to the nervous system when the nervous system faced with damage, inflammation, or even infection. And they could be change the form from deactive to reactive form. And when they become reactive, they can work efficiently and have an influence on our nervous system. For example, when the nervous system faced with infection, they change to their reactive form and do their activity. And there is another point here that when they become more reactive and this kind of reactivation prolonged for a long time, they themselves cause damage to our nervous system. As you can see here, we have a hematopoietic microglia here, and this kind of microglia helps the oligodendrocyte to be mature and have a myelination effect on our neurons and to protect our nervous system, exclusively here our central nervous system. But if their activity prolonged for a long time, their activity could be changed and they will result in damage to our nervous system. For example, these kind of cells, this is a reactive form of this cell, it's a microglia, and its prolonged activity causes that they will inhibit the maturation of the oligodendrocytes. and the activity of oligodendrocyte here is not perfect, and we faced with the problem in activity of oligodendrocyte. Then these cells are not be able to do their duties, for example, myelination of the central nervous system. And thus we will face with a damage in central nervous system when we have a prolonged activity of microglia cells, and in this case they are considered as a pro-inflammatory microglia. The microglia was they are not expanded, or they are not in a wide range, but for microglia we will focus exclusively on macroglia. As you can see here, microglias are divided into two different groups: microglia cells in the central nervous system and microglia cells in the peripheral nervous system. They are present in both of central and peripheral nervous system. And in the central nervous system, we have oligodendrocytes, as you can see here, and astrocytes here, radial glia cells, ependymal cells. They are present in the central nervous system. And the microglia in the peripheral nervous system, we have Schwan cells that are here. You can see a Schwan cell here, and the satellite cells in the peripheral nervous system. And we will talk about all of these kind of cells one by one. Okay, oligodendrocyte. As it is apparent from their name, these oligodendrocytes have a few branches. Oligo, it means a few. They have a few branches, and the oligodendrocyte in our nerve CNS are derived from OPCs. OPCs are oligodendrocyte progenitor cells. And in the development, during the development, they will be changed to the oligodendrocyte precursor cells, and then pre-myelinating cells. The oligodendrocytes that are pre-myelinating, they are not be able to myelinate the nervous system, and when they become mature, they are be able to myelinate the central nervous system neurons, and they are called myelinating oligodendrocytes. These cells form 70% of glia population in our central nervous system, and when they myelinate our neurons, they could form insulating sheaths of axons in our central nervous system, and they play important role in supporting our central nervous system. It's very important to know that oligodendrocytes are key players in neurodegenerative diseases, including multiple sclerosis or MS in our central nervous system. When these kind of cells are damaged, for example, from environmental reasons or even genetic reasons, they are not be able to produce myelin for the cells, the neuronal cells, and these cells are not normally insulated from each other, and the axons are not insulated. For this reason, when the myelin is lost, we faced with the multiple sclerosis, and you know that there are some defects in oligodendrocyte. Then the sheath and the myelin is not produced perfectly in cells, and thus it results in multiple sclerosis. It's even present in the brain or spinal cord, but it's exclusively limited to the central nervous system because the oligodendrocytes are for producing myelin in our CNS. Another type of neural glia cells are astrocytes. As it's apparent from their name, they are like stars, astrocytes. It's a Latin name that's star cells. They make contact with both capillaries and neurons in the central nervous system, and uh, also they provide nutrients and other substances for neurons. In addition, they regulate the concentration of ions and chemicals in the extracellular matrix and provide structural support for synapses because they have different and multiple branches. For this reason, they could play effective role in structural supports, and also they could be changed to the reactive form in response to many injuries or pathologies in the central nervous system, including strokes, trauma, growth of tumor, or even neurodegenerative diseases in the nervous system. And as you can see, these forms are all form of astrocyte, and as their location in the central nervous system are different, their form will be different too. You can see here we have a term which is very important in neurobiology, which is blood-brain barrier. As you may know, the blood-brain barrier is a system that protects our central nervous system from entrance of different chemicals or pathogen to our brain and spinal cord. And it helps to filter the entrance of the chemical or even cells that enter to our CNS and brain. As you can see here, it's an astrocyte with a direct connection to the blood vessel here, and it's a cross section of the blood vessel. And as you can see here, these are astrocyte projections that cover the blood vessel and make a kind of dam that do not let all the components which are present in the blood vessel enter to the brain. And as you can see here, these projections of astrocyte are completely cover the here and form the blood-brain barrier. Here we have brain, and here we have blood vessel, and these form a blood-brain barrier. And if the astrocytes faced with a problem, for example, kind of neurodegenerative diseases, or maybe as a result of some chemicals or drugs or other things, and when they damaged, they could not filter, and maybe pathogens, different cells, and other things could enter to the brain and cause different problems. This is a kind of protective effect for our brain and central nervous system that's played by astrocytes. Another group of glia cells in the central nervous system are radial glia cells. As it's obvious, the radial glia are radiated. They are long cells that are radiated in the subventricular zone of lateral ventricle. As we will talk about in next presentations, we have different ventricles in our brain, and one of them is lateral ventricle. There is a part of lateral ventricle is SVZ, or subventricular zone of the lateral ventricle, that our neural stem cells are placed there. The radial glial cells are specialized cells in the developing nervous system of all vertebrates that are placed in the lateral subventricular zone of the lateral ventricle, and they serve as scaffolds for developing neurons as they migrate to their end destination. As you can see here, it's a radial Glia cells, and these cells are playing as a scaffold and help neural stem cells to migrate to different places here, and find their final destination. And these radial cells guide cells to migrate. And these cells are newborn neurons from the ventricular zone to the mantle region. Here is a subventricular zone, and by means of radial cells, these newborn neurons could migrate to the mantle region, which is named part of neurogenesis. And as you may know that we have adult neurogenesis, and it's a part of neurogenesis process. Okay. Another group is Macroglia in CNS that are called ependymal cells. Here you can see it's ependymal cells with the different wheels here, and they are ciliated epithelial glial cells and developed from radial glia, and they cover the fluid-filled ventricle. As you may know, here it's a lateral ventricle in the brain of mouse, and here we can see it's a part of ventricle here, that the wall of this ventricle is covered as lined with ependymal cells, and here we can see that the ciliated part of these ependymal cells are toward the ventricle. These cells are responsible for production of cerebrospinal fluid in the ventricle, and they are responsible for CSF homeostasis and brain metabolism and clearance of waste from the brain, as you can see here. In the ependymal cells, we have three types of ependymal cells. These three types are called ependymocytes, tenocytes, and choroidal epithelial cells. The ependymocytes are lined with the ventricle of the brain and central canal of spinal cord, as we saw here in the last slide. These cells are ependymocytes. And tenocytes are lying the floor of the third ventricle, overlaying the median eminence of the hypothalamus. In the next presentation, we will focus on the ventricles. And choroidal epithelial cells are lying the surface of choroid plexus in the ventricles. Expect lateral ventricles. These cells were in the central nervous system, and here in our peripheral nervous system we have Schwann cells. As you may know, Schwann cells are responsible for production of the myelin in the peripheral nervous system, and they are principal glia of peripheral nervous system. We have two types of Schwann cells that are myelinating and non-myelinating. And the myelinating Schwann cells warp around axon of motor and sensory neurons to form the myelin sheaths. As you can see here, in the peripheral nervous system, it's the Schwann cells that warp around the axon of motor or sensory neurons to make it insulate. But in the central nervous system, the oligodendrocyte was responsible for making myelin for axons. You should know that here this cell is responsible, the cell body is responsible for producing myelinate. But in the central nervous system, the oligodendrocytes themselves produce some projections, and the projections are responsible for producing insulating sheaths for axons. Here you can see that Schwann cells are involved in many important aspects of peripheral nerve biology. They are important for impulse conduction, and also they are a key player in nerve development and regeneration of nervous system, and also extracellular matrix productions. When we have a defect in Schwann cells, there is a result in patient that is called Charcot-Marie-Tooth diseases. In these diseases, the patient have some problems in their muscles, skeletal muscles, and also one phenotype which is apparent among patients are their foot arc are so bigger and distinctive than normal people. And here you can see a normal Schwann cell in the peripheral nervous system that completely insulate axon. But here we have abnormal Schwann cells that are not perfectly insulate the axons. And here we have onion form. You can see like a cross section of onion. It's not completely insulating the axon. Also here we have an impaired covering of axon, and also here. These could be form of the Charcot-Marie-Tooth diseases. And another type of cells in the peripheral nervous system are satellite cells. They play as a supportive cells in the peripheral nervous system. They warp around the neural cell bodies, and they are found exclusively in the peripheral ganglia. As the sensory, parasympathetic, and sympathetic ganglia in the peripheral nervous system, and also they supply nutrients to the surrounding neurons and also have some structural functions. The glia were finished, and here we can talk about neurons. In our last presentation, we talked a bit about types of neurons. Totally, we have two classifications of neurons. One of these classifications are based on structural differences, and another one is based on functional differences. If we consider the structural differences, we have three types of neurons: bipolar neurons, unipolar neurons, and multipolar neurons. And for functional differences, we have sensory neurons, interneurons, and motor neurons. Here you can see that there are three types of neurons based on structural differences. Here it is a kind of pseudo-unipolar neuron. It means that unipolar, it means that from the cell body we have just one projections. As it is one, we say unipolar. The cell body is unipolar, and it's a kind of unipolar neuron here. And we have a multipolar neuron. Here you can hear in the soma we have different and multiple projections including dendrites and axons. For this reason we call this kind of neuron multipolar. And we have bipolar neurons here. Here we have bipolar. From one side we form a dendrites, and from another side we form axon. It's a kind of bipolar neuron. And these kind of pseudo-unipolar, multipolar, and bipolar neurons are considered as a, for example, this kind of neuron is an interneuron, and it's a kind of motor neuron, and another one sensory neuron. This classification is based on structural differences. And another classification is based on functional differences. As you can see here, their function are different. We are not group these cells by their morphology or their morphological characteristics. We say that it's a sensory neuron, interneuron, and motor neuron. It's a sensory neuron because it received stimuli and transmit impulses from our environment, from our receptor, sensory receptor, and transmit the impulse to the part of central nervous system and transmit these impulses into the interneuron. The interneurons are located in the central nervous system, while the sensory neuron is located in the peripheral nervous system. And the interneuron is responsible for transmission of impulses to the motor neuron. When their central nervous system process, it's more complicated, but in the schematic I demonstrate to be more easier to understand. When the impulses will process in the central nervous system, then the interneuron will transmit the impulses to another neuron, which is motor neuron, and conduct the impulse. And the motor neuron, which is located in the PNS, and some parts are in the CNS, are responsible for making reaction. And you can see that all of neurons are divided in three groups of motor neurons, interneurons, and sensory neurons, as they are considered for the functional differences. Thank you for your attention. If you like this video and this video was helpful for you, please put the like button and subscribe my channel for my next videos.